Is that okay? Do you see it?
Yes.
Excellent.
So Hagen, I remind you to mention that we are locking the meeting 5 minutes after it starts.
Yeah. And that we have now 3 weeks before the next webinar starts, right?
I guess so. I don't remember.
Yeah, it's I think it's March 1st. It's 3 weeks, I think.
Yes, March 1st.
Yeah, that's Jose.
Good. Yeah.
Hi, Susan.
I actually don't see all the people.
Hi, Peter.
Hello, hello, Ben. Hello, Angela.
Hello.
Nice title.
Indeed.
So you're doing a lot more solid state. Is that what we're going to hear?
Not really. Solid state is on capsid and mostly fluorine now. So you'll see a tiny bit of solid state.
Okay, I was just curious because of the title, Crystal Clear.
Ah.
Maybe you're just criticizing crystallographers. They're getting a lot of hassle lately, aren't they?
No, they don't.
Between the intrinsic disordered people and the artificial intelligentsia, I mean, it's they don't have any friends anymore.
And the electron microscopy.
I feel sorry for them.
I don't know. I should have then my other title. I have a talk that I give with a little help of my friends, which tells you that you need everybody to work together.
Yes, yes. I'm just joking. I'm sorry. I should think about the fact that when I make jokes to such a large number of people, there'll be some people who will misinterpret them. But anyway, oh well.
So the numbers are still increasing, right? So let's.
Yeah, you can wait another moment or two.
Let's wait another minute.
Our next speaker just joined.
Oh, Jose.
Hi, Jose.
Hi. Well, I think it leveled off now.
Yeah.
Right. I think we can start. Okay, so it's great seeing so many of you again joining our bi-weekly webinar in protein folding and dynamics. So welcome all of you. It gives me a great pleasure to introduce our speaker today, Angela Gronenborn. Actually, her contributions to our understanding of protein structure, function, and dynamics are so manifold that it's almost difficult to find a good start for the introduction. So actually, when she started as a postdoc, NMR was still in its infancy. So what was basically available was one-dimensional NMR, proton NMR, but still with these limited tools, she showed directly for the first time that proteins exist in alternative conformations, which at that time was a huge leap forward because it explains partially how proteins and enzymes interact with their ligands and substrates. And now since then, she was a major driving force in developing NMR spectroscopy into the versatile toolbox that we have available today. And this included development of multidimensional heteronuclear spectroscopy, particularly also the combination with restrained molecular dynamics and simulated annealing for three-dimensional structure determinations. But method development for Angela always seemed to have gone hand in hand with important biological questions that she wanted to address. And so over the years, the Gronenborn group has solved solution structures of a large number of proteins that are medically and biologically important. And this includes cytokines, chemokines, transcription factors, and of course, most prominently, HIV-related proteins. Now, a few words about her CV. Angela studied chemistry in Cologne and also received her PhD in Cologne, and then started a postdoc in London at the National Institute for Medical Research. And afterwards, she directly went on to a group leader position at the Max Planck Institute for Biochemistry at Martinsried. But already 4 years later, she left for the NIH to become head of the structural biology section at the NIH. And so finally, since 2005, she is a professor at the University of Pittsburgh. Now, given all her seminal contributions, of course, she received many awards and honors, and I can only mention a few. So Angela is a member of the National Academy of Sciences in the US. She's also a member of the Leopoldina, which is the German Science Academy. And she's also a member of the Norwegian Science Academy. She received quite a number of prizes. I just mentioned a few. She received the Wilson Award in Spectroscopy, the Ernst Prize in Magnetic Resonance, and also the Mildred Cohn Award in Biological Chemistry. And today, Angela will talk about the latest news about the interplay between crystallins and cataract and how to get a crystal clear understanding of that. But before I hand over the stage to her, I just would like to ask each of you, please mute your microphones such that we can listen to Angela's talk without any interruptions. As usual, we will have a Q&A session afterwards. So if you have a question, please use the chat window, type in question, and then I will call you one by one after the talk. Please also don't forget that we will close the session 5 minutes after start because of previous Zoom bombing that we had. So this is just a security measure, and so in case you exit the session, the webinar, then you can still watch it via the live stream on YouTube. And so I will also take this opportunity to mention our next speaker in this series, Jose Onorchik, who will talk about energy landscapes, protein sequence coevolution, and the links to chromosomes. However, the talk is not going to be in 2 weeks from now, because we have the Biophysical Society meeting on February 22nd, but in 3 weeks, so please don't forget that. And so with this, I'm done, and I'd like to hand over to Angela. Angela, stage is yours. Thanks so much for contributing today and for sharing your results with us. So I stop my screen, and Angela, you can stop yours. You can start yours.
And I hope everybody can see my screen.
It looks good. Can you click on the screen?
Okay.
Yeah, now it's perfect.
Okay. Thank you, Hagen, for this very nice and long introduction. I get 5 minutes more now for my talk. And good morning, good afternoon, good evening, wherever you are in the world listening. And I hope at the end of my talk, the title will become crystal clear to you. Let me start out with the slide you see right now on your screen. The French impressionist Claude Monet is best remembered for his iconic paintings of his garden, and in particular, the water lily pond at his house in Giverny. And let me just switch over to the laser pointer, because you hopefully can see this better. Everybody knows his water lily paintings. Now, in his 60s, like lots of people in their 60s, Monet started to develop bilateral cataracts, which eventually would affect his work substantially. In 1913, he traveled to London to consult with a German ophthalmologist, Richard Liebreich, who had been appointed chair of ophthalmology at St. Thomas's Hospital. Interestingly, Liebreich himself had a keen interest in art, and he'd written an article on the effects of eye disease on the painters Turner and Moretti. Liebreich prescribed new glasses to Monet, but highly recommended cataract surgery for the right eye, but Monet refused. By 1914, 1915, Monet began to struggle really severely, and he complained that the colors no longer had the intensity that they had for him, that the reds had become muddy, and that his paintings were getting really dark. In order to avoid choosing the wrong colors, Monet started to label his tubes of paint and kept a strict order on his palette. Light from bright sunlight really complicated things further, and it forced him to wear big straw hats when he was outside. His brush strokes had become broader and more coarse, and the paintings, like the cataracts in his eyes, became more and more brunescent. Monet was adamant to avoid surgery at all costs, but finally he had to agree. So he had surgery in his right eye, where his lens was removed in 1923. And these two paintings show you what his colors looked like when he was younger and while he was having cataract. And if you look at a painting, and I should have put one on the slide here, after he had eye cataract surgery, his paintings went back to the bright and vivid colors that he used to have before 1914. So what are cataracts? Let's start with the eye. In the eye, you have a lens, and the lens is there to focus light onto your retina. And when you have a cataract, there you have deposits in the eye lens, you have a clouded lens, and you can no longer focus the lens on your the light on your retina, and you have a lot of blurred vision. So the lens is an organ in which you have lens cells that, okay, now I have to try to get rid of my little icons here, where you have cells that are laid down from the center subsequently to the outside in the development of an organism. And the parts of the eye lens are called by ophthalmologists a nucleus. Then you have the capsule and the cortex, and outside you have these epithelial cells. And if you think about cataract in the US, it is really not a major life impediment, because almost half of the people in the US who are over 80 years old either have cataracts or had cataract surgery. But if you look worldwide, more than 25 million people have impaired vision or suffer from blindness, and the majority of that is caused by cataracts. This is particular so in China, India, sub-Saharan Africa. And although cataract surgery is an in-out procedure these days in developed countries, there are lots of people in these types of parts of the world where cataract surgery is not easily had. So it seems prudent to think about how one could develop therapies that would slow down the onset of cataract or delay the progression of the disease. And for that, we have to understand what cataract is about. So I mentioned that the eye lens is formed very early in development, and from the inside out, the lens cells, which are devoid of any organelles, fill up with proteins that are called crystallins, and they are in the lens cells at very high concentrations of 400 mg per mil, which is unheard of for proteins. Because you start out developing the eyelids early in development, the proteins that are in the lens, and especially in the nucleus of the lens, have to last a lifetime, because no organelles, no protein turnover in the lens cells. So what is in your eye has been put down when you were in utero. And as we all know, there is a process called aging of proteins. And obviously, hopefully those proteins that are laid down there can last 80 to 100 years. So the primary function of the crystallins is to contribute to the transparency of the eye lens and the refractive power. If you look at the evolutionary origin of the lens, and in particular the vertebrate lens, it appears to be a relatively recent innovation. And some people speculate that it has to do with the fact that vertebrates are predators, and you are a better predator if you have acute vision. If you look at the nearest invertebrate relative of the vertebrates, there are Histidines or Amphioxus, sea squirts or lancelets, and here you see a sea squirt, Chiona intestinalis. They have been used as model invertebrates in developmental biology and genomics. So we know all the sequences in the genomes. So a sea squirt is this solitary tunicate. It has these gelatinous bodies with cylindrical soft protrusions up to 20 centimeters long. And they have what biologists call eyes. They're not really eyes. They have cells with which they can sense some light or gravity. And in the sea squirt, there is a precursor gene that is the precursor to what are called beta gamma crystallins that has a sequence signature that is repeated in the vertebrate crystallins, and here you see a number of them in this phylogenetic tree that have the same sequence signatures. So it appears that all the vertebrate beta gamma crystallins come from this single domain ancestral protein. And I would just want to remind you that there are 500 million years between the sea squirt and the vertebrates. So where are those genes located? They're located on the human genome on all kinds of different chromosomes. And the three classes of crystallins, alpha, beta, and gamma crystallins, are not all on the same chromosomes. The gamma crystallins are mainly on chromosome 2, A, B, C, and D, but then we also have 2 gamma crystallins on chromosome 3, gamma S, and 7, gamma N. The 2 alpha crystallins are on chromosome 11 and 21, and the beta ones, again, are dispersed. The basic beta crystallins, B1, B2, B3, are mainly on chromosome 22, and the beta alpha ones, again, on different ones, 2, 17, and 22. And the classes really have been named because of a trivial property, namely the size of the proteins. Alpha crystallins are very large, multimers of the order of 500 kilodalton. The beta crystallins are dimers through octamers, 45 to 180 kilodaltons, and the gamma crystallins are mostly monomeric, 20 kilodaltons. The alpha crystallins are related to the small heat shock proteins, and they have been thought to play a role in preventing misfolded proteins to aggregate in the lens. And obviously this would benefit transparency in the lens. The beta and gamma crystallins, although they appear to be close relatives, are genetically quite distinct. All the beta crystallins have 6 exons. All the gamma crystallins have only 3 exons. The beta gamma crystallins at the amino acid level are related. The beta crystallins have extensions at the N terminus and at the C terminus, and the basic ones, the beta B crystallins, only have the beta B crystallins have extensions both at the N terminus and the C terminus, and the beta A ones only at the N terminus. In terms of structure, their structural architectures are very similar. They have domains that contain what are called Greek key motifs. Here is the Greek key that are on Greek vases, and here is the Greek key motif when you put beta strands together. Each Greek key motif is made out of roughly 40 residues, and there are these defined sequence fingerprints, and each monomeric domain, each domain is made out of 2 Greek key motifs that come together in this sort of beta sandwich with a sort of approximate dyad here. And the sheet 1 that's shown in red is made from strands 1, 2, 4, and 7, and the sheet 2 is made from strands 3, 5, 6, and 8. And the monomeric gamma crystallins contain 2 of those domains that are linked by the short linker. They also have what are called very specific structural features. Each domain contains a tyrosine corner. A tyrosine corner is formed when a hydrogen bond from a tyrosine hydroxyl forms between an amide or a carboxylate 4 amino acid, 3, 4, or 5 amino acid prior in the sequence to this tyrosine. There are 2 of those in a monomeric unit, 1 per domain, and in crystallins there are basically -4 corners. Each domain contains 2 tryptophans. In the double domain monomer, we have 4 of those. 2 are highly quenched in the folded protein, 42 and 130. And when they're excited, they can obviously transfer the energy to other side chains or the backbone. And even you get cleavage of the indole ring. And this causes photoproducts. For instance, kynurane is one of the known photoproducts that is being formed in crystallins. And some people speculate that these tryptophans are proposed to absorb UV light, and this may then cause photo damage in aging of the crystallins in the lens. Crystallographically, there are lots of different arrangements that are found for the beta-gamma crystallins. As I mentioned, they can be monomeric, where you have those 2 domains. They can be dimers, different dimers. They can be tetramers and even octamers. Now, what do we know about cataracts? Since there is no turnover in the lens, the proteins have to be stable. And we all know that aging of proteins can lead to some damage, either deamidation, glycation, oxidation, peptide bond cleavage. And all of those have been evoked as being associated with age-related cataracts. For hereditary cataracts, mutations are thought to involve thermodynamic destabilization of the proteins and that they then aggregate into amyloid-like structures. But so far, this is just speculation. And most of the models of cataragenesis propose that the polydispersed alpha crystallins act as molecular chaperones, and they prevent the formation of N-specific aggregates by binding and sequestering native crystallins or non-native crystallins into high molecular weight particles. And once their capacity to bind these aggregation-prone sort of damaged proteins is exhausted, then you would end up with particles that are light scattering, and this would cause cataract. However, this titration model has recently been challenged and put to rest in a beautiful paper by Johannes Buchner in Nature Structural and Molecular Biology. He showed that the mutated crystalline proteins are depleted in the eye lens and do not aggregate, and that the resulting changes in the protein composition alter what is thought of as higher order structure of crystallins in the eye lens, and that the aggregation of other non-mutated crystallins induce the cataracts, including that there are alpha crystallins in these aggregates. So for these congenital and early onset cataracts, people have done a lot of genetic studies, and mutations in over 30 genes have been found. About 8 to 25% of the congenital cataracts are hereditary, and generally the genetic changes are transmitted in a highly penetrant Mendelian fashion. And for the families for whom mutant genes are known, roughly 40% show mutations in the lens crystallins. But the pathogenic mechanism is really not known very well for the different mutants. Lots of them have been mapped, and let me start out by telling you something about a P23T variant that was shown to cause loss of solubility of the protein, and this one is associated with lamellar cataracts in Indian, Caucasian, and Chinese families. There have been several mutants mapped in the gamma D crystalline gene at that codon 24 position, and those changes result in either a P2T, P2V, or P2S variant of the protein. In the literature, it was reported that P23 cannot be crystallized, And therefore, we determined the NMR solution of this protein, the NMR structure of this protein in solution. We had to make a Histec protein because the protein itself is not very soluble. And here I show you just the chemical shift differences between the mutant protein for the amide resonances and the wild type protein. And obviously we did the chemical shift difference for Histec, wild type, and mutant protein. And as you can see, they're all located solely in the N-terminal domain of the protein and most pronouncedly around the site of mutation. The structure of the mutant protein is very similar to the wild type protein. The backbones are here superimposed in red and in blue. And the only area where you see some changes around where the substitution of the proline for the threonine occurs. And we see a slightly different orientation for this histidine 22 side chain. Now, NMR is very good at mapping out changes around a particular residue. And here you can see the area around the histidine 22 in the wild type and in the mutant protein. And we could map this all out by NOEs and have a very good structure around this side chain. This lets me to remind you that NMR really is very well suited to look at histidines, and in particular, look at what type of tautomeric state is present in a histidine and whether it's a non-charged or a charged histidine. This you can only do by crystallography if you have very, very high resolution structures, namely better than 0.7 of an angstrom. NMR lets you look very easily at this question. You just look at the N15 chemical shifts of the histidines in the imidazole ring, and you can immediately see whether you have an epsilon 2 or delta 1 tautomer, because you have very clear signatures for the resonances and also for the protonated form. And in the wild type protein, the histidine is in the N epsilon 2 tautomer. Not in the mutant one, it's in the N epsilon 2 tautomer, while in the wild type protein and in the mutant one, it's the delta 1 tautomer. So this you can very easily map out here, and we did that. Strangely enough, despite the fact that it was written in black and white that the protein could not be crystallized, after a few years, we also got a crystal for this protein. However, it only crystallized at pH 4.6, not at neutral pH. And since we know the pKa of the histidine is around 6, obviously histidine 22 is now in the charged form in this crystal structure. And here you see that we can very easily map out the orientation of that histidine in the crystal structure. And it now forms a hydrogen bond with a tyrosine 16 side chain, and the N-delta 1 interacts with a carbonyl group of serine 20. And again, overall, there is no difference between the wild type protein and this mutant protein. And if you just superimpose the individual domains, they're actually very, very similar, 0.43 and 0.52 angstroms for the N and the C terminal domain. So basically, we don't see any structural changes for this definitely cataract associated protein. So what has to be associated is certainly not anything to do with the overall structure. Another variant that is linked to an autosomal dominant congenital tract cataract in a Chinese family is a T2C change at position 127 in the gamma crystalline gene. And that change relates to a tryptophan to arginine change at position 42. And remember, this is one of these conserved tryptophans that are thought to play a role in UV absorption in the gamma D crystalline. Again, the mutant protein is less soluble than wild type, and it's also less stable, but it's not really that unstable, because if you look at the HSQC spectra that are superimposed here, in blue, the variant one, and in red, the wild type one, again, you see that all of the changes are located only in the N-terminal domain, and that tells you that the structures, again, have to be very similar. Here you see the superimposition of the structures. The protein was crystallized, and again, pairwise backbone values for just the NTD and the CTD are 0.36 and 0.28 angstrom. Now you would assume that if you change a tryptophan in a hydrophobic core of a protein, you would end up with lots of changes. However, that is not the case. Here you see the wild type situation. Here's your tryptophan side chain, and here's the arginine. And as you can see, the arginine fits very nicely into the core of the protein, and you only see very minor structural rearrangements of the surrounding residues. So an arginine fits very well into this packed hydrophobic core. If you look at the overall RMSD values, you see that it's slightly larger than what you see for the individual domains. And this has to do with the fact that you have a slight alteration of the angle between the two domains in the structures of roughly 9 degrees, and that then results in an overall slightly larger difference. Since there were no noticeable changes in the structure, so what else could explain maybe the loss of solubility or why it's associated with cataract formation? So we decided to look at the folding of this protein. While wild type protein unfolds in this very nice cooperative manner, and here I show you guanidinium unfolding curves, this particular variant has a clear two-step unfolding curve, where it appears that there is a highly populated stable unfolding intermediate. So we thought maybe we can find some hallmarks of this in the protein itself. We got some information from just incubating the protein with trypsin. The protein, as I told you, they're very stable proteins, the gamma D crystallins, and normally you don't get any cleavage at all by trypsin. But if you take that variant and incubate it overnight with trypsin, you actually, and then look at the spectrum the next day, you don't see any resonances of the N-terminal domain anymore. And what we found is that there is peptide bond cleavage between arginine 70 and leucine 80. So all of the resonances disappear. So can we see this already under native conditions? Most people, when they record their spectra, and I see this here, you never go down into the noise. However, if you are diligent and look very carefully at the noise level, you sometimes see small resonances of minor forms. And indeed, we saw this when we looked at this particular variant. Now we have 3 tryptophans. Remember, one of them has been exchanged for this arginine. And here are the N-epsilon resonances. And you can see for each of them, we have a very small resonance that reflects a minor species. If you look at the intensities, roughly 8 to 9%. And this is this partially unfolded species that you then see as the stable intermediate when you look at unfolding, that is already present under native conditions. And we could easily map that out by NMR. And also hydrogen deuterium exchange confirmed this finding. I'll skip through this particular variant, another variant where we have a to C change at a position where an arginine is changed to a serine in this wild type, in the wild type sequence. We looked at all of the possible properties that we could measure for this protein, and we did not find anything that was different in this particular variant, despite the fact it had been mapped to the gene in a family with cataracts. The valin 275D variant is also associated with an early onset cataract, and like the tryptophan 42 to arginine change, this is now a substitution of a hydrophobic residue for a charged residue in a more core region, not right in the core, but you can see here it faces the other half of the domain in this variant. Here also, we noted that in unfolding, we saw a very long plateau where a partially folded intermediate was present. So we thought we could characterize this partially folded, unfolded intermediate by NMR. And as you can see here, if we record an HSCC spectrum at 4.2 molar urea, you can see that we have resonances for the C-terminal domain present, but for the N-terminal domain, all the resonances are in a region that we call the unfolded region of a protein spectrum. And I've expanded this here. We can assign them all. So we have assignments for this partially unfolded intermediate and wanted to characterize its structure. To do this, we combined SACS and NMR, and in particular, measured residual dipolar couplings. And remember, those couplings tell you the orientation of the amide nitrogen proton vector with respect to the axis of the protein. For the N-terminal domain resonances, as you can see here, all of the RDCs are around 0. So they tell you that all of those vectors are averaged, and it's this conformational averaging for an unfolded chain that yields these very small RDCs, while for the C-terminal domain, we have RDCs ranging from -20 to +20, as you would expect for a stably folded domain structure. Here is the SACS curve for the folded protein. And as you can see, it agrees very well for the curve that you would expect for the natively folded structure of a gamma crystalline. And this is the curve you get in 4.2 molar urea, which is significantly different. In order to characterize this ensemble of unfolded NTD and folded CTD, we made use of the asteroids program that was developed by Martin Blacklich's lab. So we generated an initial large pool of structures where we had coordinates, random coordinates for all of the residues in the N-terminal domain. And for the C-terminal domain, we took the coordinates from the crystal structure. We then generated 5 ensemble of 200 structures that had averaged chemical shifts, nitrogen, amide, proton, C alpha ones and C beta ones, and then selected those that agreed with the experimental shifts. We then extracted phi-psi propensities from all of those and used those in a flexible Meccano algorithm in Martin's program to generate a pool of 25,000 models. And then this pool was subjected to an additional asteroid selection in which all the chemical shifts, the RDCs, and the SACS data were applied as constraints simultaneously. And this is now the ensemble of our structure where we have the N-terminal domain unfolded and the C-terminal domain folded. And this is shown here. Obviously, it is hard to characterize such an unusual ensemble. What we did is looked at the radius of gyration, and in our starting models, we have a distribution for the radius of gyration as shown in the black bars. For our final model, we have several areas in the radius of gyration where we have a slightly higher propensity of structures. For instance, some of them are slightly more compact, and then others are more extended as shown here, while this one is more like the average one from the starting one. So obviously you can't really put all of the structures superimposed, and this is basically the best representation we have for looking at this large ensemble where the N-terminal domain is unfolded. This particular variant forms aggregates that appear amorphous, and this suggested a non-amyloid structure. We did look at such a non-amyloid structure of a gamma-D crystalline for the other variant, the P23T1. Most people assume that the crystalline aggregates are amyloid-like, but I will show you they're not. We can make amyloid aggregates at very low pH, as shown here. There you see these fiber type of structures by electron microscopy, and those precipitates also exhibit the sort of hallmark characteristic Crux beta diffraction patterns of around 4.5 and 8 to 10 angstroms, whereas at an aggregate that forms at physiological conditions, pH 7, they appear amorphous-like, and they certainly do not have these characteristic X-ray powder diffraction radiance. They also don't bind thioflavin T, like is another hallmark that people use to characterize amyloid type of structures. When we took this precipitate and looked at solid state NMR, it was surprising to see a remarkably well-resolved spectrum. And I just show here two regions where we have alanine resonances and threonine resonances, carbon-carbon correlations. And as you can see, this is a very well-resolved spectrum, which you would not expect necessarily for an amyloid structure. When we then compared where those resonances, for instance, for the alanines were located, they're actually located in very similar positions where we know the chemical shifts are in solution, which tells us that the structure that is in this amorphous aggregate is much more native-like in the architecture of the domain than you would see for an amyloid structure. And we also looked at the precipitate that we get at pH 3, and certainly those resonances are not present. So this suggests that indeed in pH 3, you have an amyloid-like structure, but at neutral physiological pH, you do not get this type of structure. We believe that it is most likely that the domain architecture of the gamma-D crystalline in the aggregate stays to some degree intact, and we certainly did not see any evidence for non-native-like conformation like you would see in the beta amyloid structures. So how about some changes that occur with aging of proteins? And if you pick up any paper about cataract and crystallins, you invariably read in the introduction that deamidation has been shown to be associated with cataract formation. However, it hasn't really been shown. What has been shown is that if you take aged lenses and you look at mass spec, you see that deamidation of crystallins occurred. But unfortunately, this occurs whether you have a lens of an 80-year-old individual, whether the person has cataract or has not cataract. So yes, deamidation occurs, but there is no clear evidence that really it happens during cataract or is related to cataract. So what is deamidation? Deamidation is the removal of an NH2 group from an asparagine or a glutamine, and this happens via a cyclic intermediate, the succinimide intermediate, which then gets hydrolyzed to either aspartate or isoaspartate. So obviously, this particular product of deamidation, one can very easily mimic artificially by just making an asparagine to aspartate mutation in the protein. And we did this for gamma D crystallin. Gamma D has 7 asparagine, 3 in the N-terminal domain and 4 in the C-terminal domain. So we made all of those mutant proteins, again, looked at whether by chemical shift mapping, you could see any gross structural changes. Here you see the difference for the residues in the N-terminal domain, here in the C-terminal domain. And as you will appreciate, very, very small changes are present, mostly located around where you have the amino acid change. And here you see all of the 7-1 superimposed on the structure. The only one where you see a noticeable change, small as it is, here's around 1 ppm, is for the N33D variant. And looking at the structure, we explain why this one has maybe a little bit more changes, because the side chain of this asparagine in the wild type protein structure is positioned in between 2 loops and forms hydrogen bonds with amino acids on either side of its side chain to the loops, holding those 2 loops in place. And if you just look at the surface exposure of all of the asparagines on the gamma D crystalline, you'll notice that most of them are very well surface exposed. And it's only this N33 which shows very little surface exposure of its atoms. And that has to do with the fact that it's nicely tucked here in between those 2 loops. We set up crystallization for all of the variants. So far, 2 of them have crystallized, N124D and N146D. And again, superimposition of the structures as shown here show you that indeed the structures are very similar again. What about the stability? If you just look at thermal stability of all of the variant compared to wild type, you can see that all of them are equally stable to the wild type protein. Some are very slightly less stable and some are a little bit more stable, but basically no significant changes. So what about, since there are surface residues that we changed, any changes in the interaction in between proteins in solution? And this you can measure by what's called a diffusion interaction parameter, which is similar to the second virial coefficient, where you look at the diffusion coefficient in a concentration dependent, as dependent on concentration. And we measured from very low concentration to roughly 35 mg per mil. And the way you do that, you measure dynamic light scattering at different concentrations. And if you look at the diffusion interaction parameter for all of the variants, here are the N-terminal, the ones that located in the N-terminal domains, here in the C-terminal domain, you can see, again, they're very, very similar. They're all repulsive and no significant changes are seen. So certainly for the deamidation variants where you substitute an aspartate for an asparagine, we have no evidence that they are more prone to be associated and thereby cause maybe aggregation than the wild type proteins. So for the gamma D crystallins, we have a substantial amount of data. We have lots of structures for wild type mutant proteins, for deamidation variants. We have a structural model for this folding intermediate for one of the variants, and we have information about what happens in the aggregate of the P23T variant. So what about beta crystallins? We also looked at some of those. I mentioned that The beta B2 crystallins have two, grouped in two, the acidic ones and the basic ones, with the basic ones having extension at either both the N-terminal and the C-terminal domain. Now, there were also by now a lot of crystal structures in the PDB of beta crystallins, and here I show you structures that have been deposited for a truncated beta B1, a dimeric structure. Beta B2 is truncated at the N-terminus 2. This is a domain swapped structure. Beta B3 is shown as a trimer, and beta B4, again, a different dimer from the beta B1 one. Since my lab had a lot of experience with domain swapped structures, and we found that sometimes, and not rarely, although a domain swapped structure is found in the crystal, it may not be the structure that's in solution. We decided to look at this particular one to see whether we could see this domain swapped structure in solution as well. So first, we tested, is beta B2 crystalline dimeric in solution? And we looked at multi-angle light scattering, lots of different concentrations, and indeed from 0.2 to roughly 14 mg per mil, this protein is dimeric. Now, what kind of dimer structure did we find? We first carried out SAX studies, and we immediately found that the dimer structure that was seen in the X-ray was not compatible with the data we got. We then looked at the arrangement in the crystal lattice where this dimer structure was seen, and noted that if one would just take a dimeric unit out of the crystal tetrameric lattice, you could end up with a different dimer arrangement. So we wanted to test that. So we then looked at the SAX data, and if you look here for the full-length protein or the truncated one, and let me remind you, the crystal structure was for the truncated one. If we would take the domain swapped structure and try to fit it into our SAX envelope, it really doesn't fit. And you can see this also here, that the curves don't really superimpose very well. However, if you make this pseudo face-en-face dimer, then we get relatively good agreement with the SAX data. And we argue that this little protrusion here for the full-length protein is probably from the N-terminal tail that is missing in the crystal structure. And to confirm this notion of ours, we indeed then looked at the N-terminal truncated one, and there you can see that we get very nice agreement between the SAX data and this model where we have this pseudo face-en-face dimer. But obviously, SAX is a global parameter, and we really wanted to know, so what is really the structure like in more detail? For this, again, we used NMR, and we decided to use PREs. And we wanted to make a mixed sample where we put a paramagnetic tag in one unit and have a non-paramagnetically tagged monomer in the other unit. Now, beta B2 has two cysteines, cysteine 37 and cysteine 66. And we need to attach the MTSL to one of those cysteines. Unfortunately, when you attach it to 37, this leads to precipitation. So we used 66 to link the MTSL group. And then we changed cysteine 37 to a valine. And this was then used for making our PRE measurements. And as you know, you put a paramagnetic tag, and then you see attenuation of the peak intensities, and this will tell you what's close to the tag. So here is where we put our tag on the C-terminal domain. Protein is N15 labeled, and we tried to look where do we see the attenuation of the signals, obviously around where we attach the tag, as it should be. And then we also see some attenuation in the other domain. So we thought, oh, easy, we can now look how the two domains come together. Unfortunately, however, when you do this experiment, you cannot 100% you convince yourself that you will have only the face-en-face dimer, because very similar contacts are seen in this type of arrangement as in this type of arrangement in the either domain swapped or the face-en-face one. So now we had to become a bit more intricate, and bear with me because we had to make samples that were unique. So we had to make a sample where we have the reduced MTSL tag that is diamagnetic on an N15 sample, and we had the paramagnetic tag on a N14 sample. Then we mix those two samples together, and then we should get in a 2:1 ratio a sample where we have N15 with a non-paramagnetic tag and a N14 sample with a paramagnetic tag. And then we looked at the PREs and checked whether they were compatible either with the side-by-side swap dimer arrangement or the face-en-face arrangement. And here I show you in black the curves for the diamagnetic control sample, and in red for the one where we have the paramagnetic and diamagnetic mixed sample. And you can see for those resonances where we would expect PREs to be observable in the domain swapped arrangement, we do not see any difference in the PREs. However, if we look at residues where we should see PREs, if the protein is in a face-en-face orientation, we indeed see the attenuation of the signal intensity for the paramagnetic tagged protein. So from this, we unambiguously could decide that the non-domain swapped architecture is only compatible with our PRE data. And this is another example where in the crystal, you have a domain swap dimer, but in solution, you do not have this domain swap dimer, but you have a dimer that is a face-en-face dimer. And interestingly, there's another beta protein, the beta B1 crystallin, that has a similar arrangement like a face-en-face dimer that was seen crystallographically as well. With this, I have to thank all the people who actually did this work. It started out over 10 years ago, and Jinwon Jung, a postdoc in the lab, started with this work. And the work arose because I had a talk with Jonathan King, and he told me about crystalline proteins, and I was intrigued. And the first paper we wrote was actually a collaboration with Jonathan King's lab, and John Ting Wang and Jinwon worked together on this particular protein. A lot of the work and most of the papers that were published was carried out by Fang Ling, a graduate student in the lab. She solved crystal structures, NMR structures, and characterized a lot of the genetically encoded variants that were supposedly associated with cataract. Jennifer Boas, another graduate student, was a joint graduate student between my group and Patrick van der Wel, and she carried out the work that was associated with solid state NMR of the T23T variant of the protein, and characterized that in the precipitate, we do not have amyloid type of precipitates. Matthew did a lot of the sex, and he also carried out the structure of the partially unfolded folded intermediate. Jia Yong, a postdoc in the lab, did the work on the face-en-face dimer of the beta B2 protein, and the deamidation variants. Alex Güsemann, a postdoc in the lab, worked on with the help of Jeremy Gonzalez, who was a summer student who came over from Puerto Rico and spent the summer in the lab working with Alex. And all of the work was funded by the National Eye Institute, who supported me for the last 10 years for my work on crystallines. And I'm very grateful for their support, and even more grateful for all of the hard work of the people in my lab. And with that, I thank you for your attention. I'd be happy to answer questions.
Thank you very much, Angela, for these wonderful insights into crystallines. So we have the first question by Gilad Haran.
Yes, thank you very much, Angela. So my question is perhaps it goes back to the beginning of your talk when you mentioned that in the lens there is this enormous concentration of the crystallines. And so is it known what kind of dynamics happens there? Are they at all moving around? Are they diffusing around? Are they doing something, or are they completely in place like some kind of an amorphous arrangement that doesn't really change at all?
I wouldn't call it an amorphous arrangement. I think some of us working in the field have some ideas that it may actually be a sort of semi-ordered system, because it is really at very high concentrations. And you can easily imagine that at those concentrations, you can form something that maybe sort of, for lack of a better word, liquid crystalline-like. But it's very hard to look at that carefully, although this is really what I'm very interested in doing right now. We try to get at the information of just looking at very, very high concentration samples in the test tube, labeling one protein and putting very high concentrations of the other proteins in. But that's not good enough. So what we are now doing, we're actually starting to look at monkey lenses and trying to look at the lenses. And we've put the first lens into a rotor and looked at the solid state spectra, because I think this is something where we want to get closer to the real in vivo situation, and hopefully eventually we'll get there. And what was interesting, Johannes Buchner in his paper, when he looked at some of the crystallines, he did some sacs on intact lenses, but mouse lenses. So I'm trying to go to higher order lenses, not cow lenses, but human ones you can't get anymore. I tried very hard, because with cataract surgery now, they don't take intact lenses anymore out. But from monkeys, I can get them, and from also marmosets, I can get them.
Because you mentioned that at least one of the crystallines is homologous, I think, to a chaperone.
The alpha crystallines are these sort of polydisperse associates that are related to small heat shock protein, but they're very, very polydisperse. And there are people who are doing some solid state NMR on those ones, but it's very hard to do. And you always have to look at the cryo-EM, and you have to do the binning, because you never get a unique oligomeric arrangement. So you always have these multiple arrangements of these alpha crystallines.
So it's not known if they have a similar
they're
job in the eye as well.
They are beta strand proteins, that's for sure.
Okay, thank you.
Okay, so next question is by Ben.
Thanks very much, Angela. Wonderful to see this system again. I remember some early work in Rainer Janicke's lab a long time ago.
Right.
Still so much to discover on crystallines. It's fantastic. I have a detailed question and a more general question. The detailed question is, in one of the variants, the W42R variant, you mentioned that the N-terminal domain, if I understood correctly, disappears from the NMR spectrum. if you do the digest, I think.
Yes.
Right? So I was wondering, is that because it unfolds and disappears into the unfolded region, or does it actually
No, it actually it first gets cleaved at this particular amide bond at position 80, but then it gets degraded.
Oh, it gets degraded completely. Oh, okay. Mm-hmm. All right.
For the other one, where we looked at the 4.2 molar solution, where we have the completely unfolded N-terminal domain present, we see all the resonances.
Okay, yeah. Mm-hmm. And then I had a general question regarding the tryptophans that you mentioned at the beginning, because I also remember a discussion with John King a long time ago, and I think one of his hypotheses was that these tryptophans are specifically in place to reduce photo damage to the eye lens, because you would absorb the UV very efficiently and then dissipate it before it can do any harm. Is that still the current view?
I don't know. I think they can definitely absorb, but whether they can dissipate without any harm, I'm not sure, because you do see UV-induced damage of the crystallins. And I didn't put that slide in. We actually looked at what happens when you expose, for instance, the wild type crystalline to UV. And I wish I would have had my slides still up. We then saw that the one tryptophan where you see the largest drop in intensity of the epsilon resonance, which means that you do something to that tryptophan ring, is tryptophan 42. Mm-hmm. So what you then see is also you get cleavage like you see with when you have the mutant. And what we deduce from this, that the tryptophan 42-R mutant does something similar to what would happen if you had that photo damage to that particular tryptophan, which is the one that got photo damaged the most, the earliest.
Okay. Mm-hmm. Thanks very much.
Okay, so the next question is by Raquel Lieberman.
Hi, this is a really fascinating talk, and thank you so much for it. I was just wondering, and it kind of follows up on what we were just talking about, if you've ever looked at the dynamics of the wild type gamma crystalline and whether there was any dynamics already within the wild type protein that you then see when in some of these mutants.
We did that because our lab normally do all kinds of relaxation measurements, and that's why it's not reported. There was nothing of interest that we saw, unfortunately.
Well, that makes sense, I guess, in retrospect. But okay, that's great. Thank you.
Okay, so the next question is by Kirsten Lumpy.
Oh, Kirsten, yes, hello.
Kirsten.
Hi, Angela.
Hi.
Talk. Sorry, my question was in the chat, so I wasn't expecting to. But any evidence? So I'm just gonna say, I think deamidation could still have a role in cataracts. So let's just get it out there, and surface. So I would say from the take home from your talk for me is that, sure, you're working at maybe 10 mg per mil, right? But once you go in vivo, you might see at that high a concentration. So you're not seeing huge structural changes, but you could definitely be seeing interactions, protein-protein interactions, right? Kind of like Buchner's paper just showed. So it might not be the deamidation having a direct effect on the protein because you're not seeing that, but it could have an effect with a, more heterooligomer interactions. So what do you think about that? And then remember, these crystallins are really very dynamic, right? So maybe beta B2 formed a domain swapped in the crystal structure, but it could go back and forth between the non-domain and the domain swapped in vivo. And again, Do you have any evidence for that, or what do you think about that idea?
Obviously, it's very hard to disprove something where you don't have any data on. So we've looked very carefully at very high concentrations, and we've made samples where we have, let's say, gamma labeled and the beta not labeled. Nobody has any in vitro system where we have the composition that we have in the eye lens. So this we can only really follow in the eye lens. And obviously there can be things happening in the eye lens that we cannot at the moment reproduce in the test tube. But for the protein-protein interaction, that's where we very carefully, at least for the gamma D deamidated proteins, went to high concentration and measured the interaction parameter. And there we did not see any difference, at least for those deamidated variants. That's all. I can only have information on what I measure. Everything else is still possible.
Well, if your in vivo whole lens model will be really interesting. That's very cutting edge and very exciting. Thank you.
Okay, the next question is by Miranda Lynch. Miranda?
Yes, thank you. That was a absolutely fantastic presentation. And my question was also about the deamidation. You know, you mentioned that it obviously can turn into multiple species, but you really only can measure the one with the aspartate. Any speculation on whether, first of all, whether there's any preference for exactly what forms it assumes and whether that would have any consequences?
For peptides, you can measure it. At low pH, you can get easily isoasps. Mm-hmm. And those deamidations, I've unfortunately worked on proteins all my life, and I've seen deamidations happening in other proteins when you keep an NMR sample for a long time, and when you have a gly-asp bond, you can get them much more easily. If it's in a loop, then it's in some other structure. And I even had resonances that come from isoasp in other proteins, not in the crystallins. So this is definitely a mechanism that does happen. Unfortunately, I wish I would know how to put isoasps into proteins. I talked with people who evolve the non-synonymous tRNA synthetase pairs, and we can't put isoasp in. You can only do it in peptides, and I would rather have it in the real life protein. So right now we are stuck with what we can do chemically or biosynthetically.
Great, thanks.
Okay, so I'm not sure whether this is a question, but I think so. So Yusra Kauder, he gave a statement, but I think, or she, but I think there is also a question associated to it. So if you want to unmute yourself.
I didn't have any question. I was just saying, when you mentioned the small heat shock protein is what I work with, I know that it's constantly carrying out subunit exchange depending on several conditions.
Yeah, certainly the alpha ones do that. The beta ones don't, and the gamma ones don't. And also following up on Kristin Lampis, certainly there is no exchange between domain swapped and non-swapped for the beta B2 we looked at in solution. It's definitely not domain swapped. It's just a Fas-on-Fas dimer. It is a dimer, but a Fas-on-Fas one.
So then the next question would be by Ruth McTiernan.
Hello. Not sure if this is working.
Yes, it is.
It's working.
All right. Okay. I was just wondering about the situation where the crystallins are near the membrane. Do you know if does the oxidative damage to crystallins over time affect their interactions with fiber cell membranes, or the other way around?
That I don't know, and certainly we only know what people or ophthalmologists have observed in cataract, because some cataracts are nuclear, which are very much in the center of the lens. Then you have capsular ones, you have peripheral ones. And I didn't mention that one of the crystalline variants that are associated with cataracts, it has been reported that in the lens, they've observed little crystals of proteins, but I don't know which of the crystallins were the crystals from.
Right, thank you. I think that might be Cry-AB. I'm not, or Alpha, one of the ABs. I'm not sure.
Yeah, yeah, it was a beta one. No, I don't know. I can't remember. I have to look it up again.
Yeah, I've only looked at the membrane side.
Okay.
Thank you.
Okay, so I don't see any other questions in the chat, which gives me the chance to ask the final question. Angela, I would like to go back to the gamma D crystalline. And so for the W42R variant, it was very clear that you formed this intermediate, which is 8 to 9% populated under native conditions. Yet for the V75D variant, the intermediate was populated at quite high urea concentrations. So I would guess it's really tremendously underrepresented under native conditions. So how can it have an impact on cataract if it's basically not abundant at all?
This is my pet peeve with physicians when they say they do GWAS analysis all the time of genes, right? And you see for all kinds of conditions that this particular condition, disease, is caused by the change in whatever gene. And I always say, how can you say it's caused by? It's associated with. We really...
Right, that's the difference between correlation and causality.
Exactly.
Okay. All right, so there are no further questions, but a lot of thank you comments from David Nesbitt and Miranda and many other people. So thanks a lot, Angela. It was great to have you, and yeah, have a great day, and hopefully watch a few other webinars in this series.
And thank you for having me, even if I'm not a card-carrying folder in your books.
Oh, well, I think that was the reason why we had you. Okay, everybody.
Bye.